Welcome, everybody. Oh, can you hear me? Very good. Okay. Welcome, everybody. Please take your seats. We're about to get started here. I'm Josh Tenenbaum, and it's my great pleasure and honor to introduce our next speaker, Nancy Kamwisher. Nancy has received many honors in her career, including the Troland Award from the National Academy of Sciences in the US, the Golden Brain Prize, it actually is golden, and the Carvalho-Heineken Prize, and that's why she's giving this lecture here in honor of that. In addition to her, she's a member of the US National Academy of Sciences, the American Academy of Arts and Sciences, and in addition to her many scholarly achievements and impacts, her work has had quite an impact on the public and has quite strong public exposure. Her TED Talk and other brain talks have been viewed well over 1 million times. My favorite example of Nancy's public outreach is her cameo appearance in the last James Bond movie. Technically, it's not she who has a cameo appearance, but rather the fusiform face area, or FFA, the first functionally specific brain area that Nancy discovered and named with her colleagues. And it appears in the movie at a key moment in the climax. Sorry for the spoilers, but if you're a real Bond fan, you've already seen it, I'm sure. The arch Bond villain, Ernst Stavro Blofeld, uses this as the means to wipe all of Bond's memories and of all the people he loves, and thus take away his humanity by making a very targeted micro-lesion in his FFA. At least that's the plan. But this appears at the classic moment of every Bond movie, the "before I kill you, Mr. Bond" moment, where Blofeld decides to give a brief mini-lecture on how the FFA works, which provides just enough time for Bond to formulate his escape plan. Wonderful, you should all see it. There's even fMRI images to go with it. But there's so many things that are wonderful and accomplishments of Nancy that I can't give all of them to you. I'll just give two, I think, personal perspectives. One is, I think Nancy Kamwisher deserves more credit and responsibility than anyone for making the study of functional brain imaging a true part of cognitive science by bringing the theoretical depth, the conceptual clarity, the methodological rigor, and the constant sense of innovation that we most prize in our field to the study of the human brain and how the mind works in the brain. And you're going to see some of the latest methodological advances, I think, even at the end of her talk. It might be a surprise for some of you who've seen her talk before. The other thing I would say, just closer to home, is Nancy deserves more credit than anyone for making my local community of cognitive science at MIT the place that it is, for making it both an incredibly supportive and nurturing environment, and also to have the highest standards of scholarship and intellectual rigor. And this is an approach that she's cultivated in her own lab and spread through her many students and postdocs and their students and postdocs, really to our entire field. So I really, truly, genuinely feel how lucky I am to have Nancy Kamwisher as my colleague and my great friend. I think we're all very lucky to have her in our field, and we, right here in this room, are all incredibly lucky to be seeing her talk for the next hour. So let me turn it over to Nancy.
Hi. Thank you so much, Josh. It's incredibly sweet. It's me who's lucky to have Josh as a colleague. I want to start this talk with some good news, with a broad overview of what I think is real progress that we've made in our field over the last 20 years. And then I'll go on to talk about what I think are some of the big open questions that I and others are trying to approach now. So let's start with the overview. To appreciate the progress, it helps to step back to where the field stood circa 1990, before my story begins. And back then, from the study of patients with focal brain damage, we knew about the approximate locations in the brain of regions involved in perceiving faces, in understanding language, and directing attention. But that was kind of it. And importantly, that could have been it. That is, it wasn't at all obvious in 1990, I still remember, that there was much more fine-grained functional organization to be discovered in the brain. But then functional MRI came along, and here's where we are today. So there are now dozens of regions of the cortex for which we have a pretty good idea of what they do. And each of these regions is present in approximately the same location in pretty much every normal person. And This is, of course, a very highly schematic diagram. There aren't neat little colored ovals in the brain. So let's look at some real data for any of you who don't do this kind of stuff to show you what I mean by this diagram. So here's the bottom of the hemisphere, bottom view of the brain in one subject, showing the right hemisphere on the right, left hemisphere on the left, and the outer parts are the back of the brain, inner parts are the front of the brain. This is an inflated cortical surface, so you can see the whole cortical surface. And out there on the bottom of the brain is the little part that features in the James Bond film. That's the fusiform face area in blue. And what I'm showing you in this bar chart is the much higher response of that region to faces in the blue bar than to a whole bunch of other kinds of categories. That's what I mean by saying that region responds selectively to faces. And if you're unimpressed by the selectivity, I'm a little bit unimpressed too. I'll show you some better data. With better methods, you can see that it really is much more selective than that. Right next door is another region right here that responds selectively to scenes. Here's the response to scenes in the green bar and to other categories in there. Over in the left hemisphere is a particularly interesting region, that little yellow blob that responds selectively to words and letter strings presented visually. That's called the visual word form area, and that region is of particular interest because it's the one patch of cortex for which we know that its functional specificity is trained up by experience, because kids don't have it until after they learn to read, and as adults, it responds strongly only to orthographies you know, not to orthographies you don't. And we'll get back to it and its origins shortly. Out on the lateral surface of the brain, there's some other good stuff. That little purple blob responds selectively to bodies. That's a purple bar right there. And over in auditory cortex in the left hemisphere, that orange stuff, that's a region that responds selectively to the sounds of speech. And importantly, I mean the sounds of speech, not the meaning of language. That region responds equally when you listen to speech in a language you know versus a language you don't. It's not about the meaning of the sentence, it's about the difference between a ba and a pa. Now, one question that I thought years ago is maybe we have these special bits of brain only for aspects of perception. After all, Marr and others have noted that perception is composed of a bunch of kind of different computations, and you might think it would make sense to decompose it into parts. So do we have this for maybe higher level aspects of cognition? Well, indeed, here are some of the classic language regions here in red, showing that they respond strongly when you read or listen to a sentence compared to lots of other mental activities. And so a few years ago, Ev Fedorenko collaborated with me in asking whether the language regions are really specialized for language per se, or whether they're also engaged in lots of other mental functions. So what she did was to scan subjects doing lots and lots of other tasks, the kinds of things that people have argued overlap with language in the brain. Things like mental arithmetic, working memory, appreciating music, etc. So the details don't matter, just lots of other tasks that you might have thought would overlap with language in the brain. And so then what she does is she identifies those regions with localizer tasks, in which you get a higher response reading sentences than reading non-word strings, sentences in black, non-word strings in gray. And here's the response to all those other conditions. In other words, those regions really just don't respond much at all to any of those other tasks. Lots of other brain regions do, often right next door, but not the language regions. Same deal for frontal language regions, roughly Broca's area. And I particularly love this finding because to me it's not just another selective blob in the brain, much as I do love those, but it's an answer to the long-standing question about the relationship between thought and language. So probably every one of you, before you even became a cognitive scientist, wondered, what is the relationship between thought and language? Can you think without language? Are they the same thing? What's up with that? And I think these data start to answer that question. They tell us that language is really distinct from most or all other aspects of thought in the brain. And by the way, even stronger data comes not from us, but from Rosemary Varley in England, who has been for 20 years testing global aphasics who have essentially no language ability in either production or comprehension. And she has shown one by one that those people can do every task that you might have thought would require language. They can do arithmetic, they appreciate music, they can do logic problems, they can navigate, even the aspects of navigation that Liz Spelke thinks require language, they can do, and they can think about each other's thoughts. So language and thought are really not the same thing. Okay. Perhaps the most astonishing to me patch of cortex comes from the work of Rebecca Sachs. It's this little pink blob right there, right about here on the lateral surface. And that region responds only when you think about what other people are thinking. And what's cool about Rebecca's work on this region is that over a decade of work systematically testing this region, she showed how remarkably selective it is. It doesn't respond when you think about other people's appearance, their kind of weird cultural practices, very engaging to read about, but not involving their minds, doesn't activate that region. Even reading about another person's bodily sensations like thirst or hunger or pain don't activate these regions. It's only when you think about another person's thoughts. Now, not all regions of cortex respond to some highly specific mental function. Indeed, these white bits out here have been named the multiple demand regions by John Duncan because they respond to multiple different kinds of cognitive demand. And so, for example, here are a whole bunch of very different kinds of tasks. The details don't matter. The solid bars are the difficult version of a task, and the light bars are the easy version of the same task. And you can see across lots of different kinds of tasks, those same regions are engaged. In some ways, those regions are a bigger, deeper mystery than any of the functionally specific regions that I've been focusing on. Okay, so that's my whirlwind overview of functional specificity in the cortex. And I think it amounts to, real progress that we've made, that each of these functionally distinctive regions is present in pretty much everyone, including regions specialized for some of the most quintessentially human abilities, like music and understanding other minds and language. And I like this diagram because I like to think of it as a kind of sketch of who we are as thinkers. But at the same time, this is just the barest beginning. And I think really the way to take this diagram is as a roadmap for future research. So what I want to do in the rest of this talk, messed up my pop-ups, is consider three next questions, okay? And the first next question is, really? These regions just do one thing? I do realize that most of my field thinks I'm a bit of a nut for insisting on this extreme view, but I'm going to show you some data and show you why I think it's true. So let's do that by taking on what I consider to be the most important challenge to this view. And that's a challenge that comes from a method called multiple voxel pattern analysis. And if you're not a functional MRI person, don't worry about all the acronyms. It just means that the basic idea that starts with Jim Haxby and then goes on through Frank Tong and Kamatani and pretty much everyone else in the field now, is that instead of just measuring the magnitude of response of a region, which is all I've talked about so far, we should look at what information is contained in the pattern of response across voxels within that region. And the idea is if you can read out from that pattern of response some other kind of information, that's what we care about. We should care about information in the brain, not activation. And I think Haxby's absolutely right about that. As cognitive scientists, we should use the brain to look at information, not just activation. So why is this a serious challenge? Well, Haxby showed way back in 2001 that if you look across the ventral visual pathway, there are very distinctive patterns of response for different objects. And more relevantly to my claims, if you look just in the fusiform face area, you can read out a little bit above chance whether the person was, from the pattern of response, whether the person was looking at, say, shoes or car or chairs, okay? And that's an important challenge because it says there's information in there about other stuff beyond faces, okay? So I think that's the most important challenge that's been raised, so I'm going to take a little bit of time to grapple with it. And the first way I want to grapple with it is to point out that actually, even if we had an idealized, somehow ideal model of the perfect face recognition system, wouldn't it also be able to For example, if you imagine taking a shoe and projecting it into face space versus taking a chair and projecting it into face space, wouldn't you end up with two different representations, right? And if so, then the existence of that information in the fusiform face area wouldn't count as evidence against the idea that it's really a specialized face processor. Okay, so how would we test this? Well, one way to test it is to use a convolutional neural network trained only on face identification as a proxy for an ideal face recognition system. So my postdoc, Katarina Dobbs, has been doing just that. She took AlexNet, which is the kind of famous network that kind of relaunched all the excitement about deep nets back in 2012. This is the architecture of AlexNet. And she trained it on a huge database of faces. This is a database that has close to 10 million face images in it, and it has hundreds of images for each individual identity. So Katarina trained it on 1,000 identities with many different images of each. And so then she looked at its ability to classify faces at the end. Now, the important thing about this network is it has only ever seen faces, and it has only been trained on face recognition. And after training, it's actually very good at recognizing novel images of the trained faces. It's close to 100% accurate. And this is akin to the things you guys all have on your cell phones and on Facebook. I don't do Facebook, but I gather the images get labeled with the correct names, mostly. It's this kind of rigmarole. So now the key question is, now that we have this idealized face recognition system, can it discriminate objects above chance? Well, so the way you do that is to find out whether the features in the penultimate layer can decode objects. So you feed objects into this network, you throw away the face classifier, and you pull out the features from the penultimate layer, and you use them to train a linear support vector machine decoder. That's just a way to ask whether the information is extractable from that representation at the top of the network. And what you find is, indeed, that classifier performs above chance at decoding objects. Chance is the dotted line down here, and that's above chance performance. So what that shows is, yes, indeed, a network trained only on faces can, in fact, discriminate objects a little. Okay? After all, the fusiform face area can only do it a little, too. So that means that above chance object recognition doesn't mean the system is not optimized all and only for face recognition. Same for the FFA. But the real question is the FFA used for object recognition? Is that information about non-faces ever read out by the rest of the brain? And for that, we need a causal test. So I got a phone call a few years ago from a colleague who told me that this guy right here was in Japan awaiting neurosurgery, and that they had already installed electrodes all over the bottom surface of his brain, as you can see here. And if you don't know your way around the brain, it doesn't matter. Here, for comparison, is the bottom of my brain, showing in red my face selective regions. And you can see by analogy that those electrodes seem to be placed right over the good stuff. So did I want to collaborate? Duh. So we quickly made some stimuli and sent them to Japan, and this patient very kindly agreed to look at the stimuli and allow us to record from his brain. And so what we see is, for example, in these electrodes right here that seem to be in an analogous location to my fusiform face area, here is an extremely selective response. So this is a response of that one electrode. This is time on the x-axis, response to faces in red and all other categories down here. So when I mentioned earlier that the selectivity you see with functional MRI is a little bit meh, that's because functional MRI is a blood flow measure. It blurs the hell out of the data. But if you actually have electrodes right on the surface of the brain, you can get really beautiful selective responses like this. But really the point of all of this is we were able to do a causal test, because the neurosurgeons often electrically stimulate a part of the brain in order to map out functions before surgery. And so what I'm going to show you is a causal test of the function of this region, in particular the test of whether it's causally involved not just in face perception, but in the perception of non-face objects, by asking how electrical stimulation of that region affects the patient's percept. So here is our movie. And importantly, this patient doesn't know about the fusiform face area, and he doesn't know where his electrodes are. You can see him getting stimulated right there. You can watch.
全部
そうです。
全く違う人なんだ。
違う人になります。
どんな感じの人でしょ
髪が変わってないんですけど、目、言葉であれするですね。もう一回やって。
う。もう一回。
One more time.
One more time.
この部分だけなんかアニメでできそうな感じのキャラクターの顔みたいに変わったんですけど、
はい。
口はそんなに変わってなかったんですけど、鼻目こんな感じでした。
Okay, so that shows the causal role of that region in face perception. The question here is, what is its causal role in object recognition? Does it affect the percept of an object?
そんなに変わらない。
No change.
最初の1秒ぐらい始まって1秒ぐらいがなんか横になんか目口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら気が付いたらもうこの箱にしか見えてないっていうか、なんですかまたさっきみたいに横にその目と口って感じでちょっと
アイス。
This is a kanji character. It's foreshortened, so it's hard to see.
ごめんなさい、もう一
もう
回。
一回。One more, please. One more time.
やっぱり顔に見えましたね。目口らへんって感じ。
Okay, you get the idea. So I think it's not much data. We have about an hour of it, and this was replicated again and again, all up and down, all the face selective sites produced more or less the same result, and the other ones did not. And I think it's not just causal evidence that region is involved in face perception, but it suggests that even though there's information in there about other things besides faces, apparently disrupting that region does not disrupt your percept of those stimuli, other than to put a face on top. Now, you might wonder, faces are special in many ways, so maybe anything you do just makes people see faces. We see faces in clouds. It's just not hard to get people to see faces. So what happens if we look next door in electrodes right there, which are in a position corresponding to a region in my brain and other brains that respond preferentially to color. And in fact, in him too, we found a preferential response to color in that region. So let's see what happens when those next door electrodes were stimulated.
"この箱の真ん中見てたら、この半分ぐらいがなんか虹色になんか光ったりしてますね。この真ん中見てたら、ボール見てたらもうやっぱ先の明らかに幅広い感じ、なんかちょっと虹色に点滅してるんですけど。先生のやっぱ鼻あたりに目線見てたらやっぱこちらの顔半分が同じような感じで半分が変わる。
Of course, he's showing that he sees the colors on the left side of everything. He doesn't know why that is, but we do. It's because he's being stimulated on the right, and the color regions are contralateral. So really? Yes, really. Some regions of brain really are very selective for just one thing, at least for the fusiform face area, and although the evidence isn't quite as strong for some of these other regions, it's pretty strong. And so I think what that means is that even though there is non-preferred information about non-faces in the fusiform face area, one, we would expect that anyway, even of an idealized face processor, and two, so that's not evidence against the specificity of the fusiform face area, but two, it doesn't seem to be read out other than as a face on top of anything else. Okay, so having made the case that we really do have some regions that are highly functionally specified in the brain, I wanted to move on to consider a second, quite different question, and that is, how does all this systematic structure get wired up in development? I find it impossible to look at a diagram like this and not wonder, how do you build brains so that they all end up with very similar structure, down to this incredible level of detail? Now, spoiler alert, I'm not really going to answer that, but I'm going to show you some cool data en route to not totally answering it. So the first question you might think of is, okay, when does all this organization arise? And lots of people, starting with Colonique Gross-Spector and many other people since then, have been scanning kids age 5 and up, some studies down to age 4 and even 3 years old, and finding that some of that organization seems to be there. But even those ages are really late. We need to go earlier. And that's almost impossible. But my amazing colleague, Rebecca Sachs, spent about 5 years trying to figure out how to scan awake infants. And she did all kinds of things, including getting dedicated, specialized infant coils made. And actually, one of the most extreme things she did was to go produce her own subject. So here's Rebecca and Arthur, the champ of her first paper. I don't know how many times Arthur was scanned in his first 8 months of life, but lots. And Ben Dean and Hillary Richardson, who participated in this study. And it was well worth the 5-year effort, because what Rebecca showed is that a lot of the same structure of face and scene representations are present by 6 months of age. So here's an infant brain, and here for comparison is adult brain. And you can see the similarity in the location of activations for faces and scenes at this age. So that's hugely important. It pushes back tenfold the age by which the stuff has to get wired up. It says the spatial organization is already there by 6 months. Importantly, those selectivities are not yet adult-like at that age. It's still developing, but at least the kind of cortical layout has already been specified to some degree by that age. But of course, we want to know not just when, but how all this stuff gets wired up. And so recently, we've been taking a whack at this question for the case of the fusiform face area with my postdoc, Ratan Murthy, shown here. And so we've been considering two questions. First, how does selectivity for faces arise in development? And second, first, let's consider the standard hypothesis that is in the field and that most people would come up with just based on kind of common sense. And that is, presumably, a big chunk of face selectivity is wired up from our extensive visual experience with faces. After all, infants look at faces pretty much from birth. They look at them a lot, as Linda Smith's beautiful data show. We get loads of experience with faces. Even stronger evidence comes from a very cool study published recently, the title of which is Seeing Faces is Necessary for Face Domain Formation. This was a study in monkeys who also have face selective patches like humans do. But these guys raised baby monkeys without ever letting them see faces. They got plenty of visual experience, plenty of primate cuddling and so forth. They were raised by human caregivers who had welder's masks over their faces at all times. And those baby monkeys did not form face selective patches. Hence the title, Seeing Faces is Necessary for Face Domain Formation. Now that seems like the data are very strong, the conclusion seems pretty solid. But around the same time, another paper was published with pretty much the opposite title. Its title was Development of Visual Category Selectivity in the Ventral Visual Cortex Does Not Require Visual Experience. And this was a paper by Van den Herk and Hans Optebeck and others. And what they had done was to scan congenitally blind subjects listening to different sounds. And they found weak but real preferential responses to face-related sounds, like chewing and sighing and kissing, compared to scene-related sounds like traffic and waves and stuff like that, in roughly similar regions. So now we're at a puzzle, and we don't really know what the role of visual experience is, after all. Second question is, how does face selectivity know where to go in the brain? How come it always lands in the same place? What is up with that? Right? It lands right there all the time. Well, one of the standard hypotheses is that we foveate faces, and it's well known that there's a lot of input from foveal retinotopic cortex straight into that patch. It has a foveal bias, and maybe what that does is basically pipe faces right into that patch of cortex, which then gets trained up by all that face experience. So that's been a much articulated and very reasonable hypothesis. Another related but different idea is that maybe the cortex starts early on at birth or soon thereafter with some kind of proto-visual feature map. And maybe there's an early developing bias for curvy things right in that patch of cortex, and then faces land there because they have curvy features, and experience trains it up from there. So those are some of the ideas that are out there and that seem eminently plausible. But our approach was to take another whack at this using haptic stimuli in congenitally blind subjects. So what we did was to scan congenitally blind subjects while they haptically explored stimuli like these here. So these are little 3D printed objects, yay big. And the way it works is we have the extremely high-tech device of a disc of cardboard sitting on the subject's belly. And we have a postdoc posted just immediately outside the scanner bore, rotating the cardboard at even intervals, so that the subject's hand can be placed in one location. Motion artifact's a problem, so they put their hand in one place, and the stimuli move underneath, enabling them to haptically explore, it's a little bit slower than this, each object in turn. And in that way, we can do a functional MRI experiment looking for haptic face selectivity in blind subjects. We also ran control-sighted subjects looking at visual versions of these stimuli, kind of like these rotating ones up here. Okay? So that's the experiment. What do we see? Let's look at the sighted subject data first. We see, here are two example subjects. We see face-selective responses, this is the ventral surface of the brain, in the fusiform gyrus, in the usual locations, nice and robust, in sighted subjects, as expected. And if you look at the overlap map across subjects to see where this lands in general, you see that it lands in the usual location, importantly, just lateral to the mid-fusiform sulcus, shown in white here, this tiny little sulcus that no one ever noticed until Kevin Wiener and Kalanick Grospector pointed out that it was a systematic landmark and that face selectivity always lands on the lateral side of it. And so we replicate that here. What about the blind subjects? Well, we also see face-selective responses in blind subjects, and it looks pretty similar in a similar location. And here it is in two of our blind subjects. Now, importantly, we see it very clearly in many of the subjects, most of them, but not all. It's less consistent in the blind subjects than the sighted subjects, and we still don't know why. But if you look at the overlap map in the blind subjects, you see it's in the same anatomical location, also just lateral to the mid-fusiform sulcus. Turning to the selectivity of these regions, one of the things that I consider a key hallmark, the reason I pay attention to these things, that they're whopping effects. If they were minuscule effects, I wouldn't care. But here's what you see, the standard kind of finding for visual responses in sighted subjects, response to faces in red, and the other conditions in the other colors here, showing a nearly 2-to-1 response selectivity for the sighted subjects. And in the blind subjects, we see an extremely similar response profile, nearly 2-to-1 selectivity for faces, even though here we're including all of the subjects here, even the ones who didn't show it. So one important thing is the absolute magnitude of response is higher for visual presentation in sighted subjects than for haptics in blind subjects. But other than that, it really looks like seeing faces is not necessary for the development of face selectivity in the fusiform gyrus. So we've ruled out that hypothesis. But what does that mean? Does that mean that face selectivity is innate? Not necessarily. Blind people do have some haptic experience with faces, certainly their own faces, maybe other faces, especially when they're babies. But importantly, blind people don't feel faces that much. When we first started doing this work, I Googled it, and there are all these Google pages of blind people chatting about how much they hate it when they say, like, why are sighted people always asking if we want to feel their face? That's disgusting. So it's like, it's not a thing, right? So surely there is some haptic experience, but it's not like frequent. And certainly there's no haptic expertise for face perception in congenitally blind subjects. So why does this activation land in this systematic location? Well, it's really not likely that it's due to a foveal bias of that region because blind subjects don't have any foveal vision. So that story doesn't really make sense. What about this other hypothesis that there's some proto-map for visual features? Well, visual features won't cut it, but we could slightly change that and say, okay, shape features. Maybe they're amodal shape features, and maybe through touch you can access some of those same shape features. There's actually quite a literature suggesting that. So maybe that location is the location that responds to curvy things, whether you see them or feel them. Okay? So we decided to bring our blind subjects back for a second session and test them to see whether they show a curvature bias for haptic stimuli. So we printed out little spheroids and cuboids and reran the same experiment, including our old conditions. And this was particularly nice because we could use their face selective regions defined from the first scanning session to nail down exactly where we'd look in the second session, eliminating degrees of freedom, which is always a good thing, enabling us to replicate our earlier results. And so here are the new data from the same subjects, nicely replicating the face selectivity in a second session. So that's cool. But what about the response to spheroids and cuboids? No higher for spheroids and cuboids, and really low to both. So no evidence there for a curvature bias. But also another way we can test whether it's really maybe we tested the wrong kind of curvature, right? So another way to test whether that region is really about some kind of amodal shape, maybe some other proto-shape feature, is to go back to the Vandenherk study that I mentioned that reported selective responses to auditory stimuli. So we got their stimuli and replicated their experiment, which I also wanted to do because even though I know, the lead author, Hans Optebeck, he's amazing, and I would trust any of his data, with my life, nearly. I wouldn't trust my own with my life. I still wanted to replicate it because it just was so surprising. But here it is, it replicates. Here's the response to face-related sounds in that same region, and sounds related to scenes and bodies. That's stuff like clapping, footsteps, knuckle cracking, etc. So wow, that's amazing. And that suggests that something more than just shape is going on in this region. Okay, so it doesn't seem like a general preference for curved features can really be the whole story of why face selectivity lands in that patch. So much for that. So now we're in a pickle. We've ruled out all the standard and sensible hypotheses, so what's left? Well, there's another hypothesis that's actually been out there for a long time in various guises, and that is one based on connectivity of the brain. And so this is based on a very old idea, starting with primate work decades ago from Passingham and Van Hessen and others, in which they showed in monkeys that functionally distinct patches of cortex have very different connectivity to other remote brain locations. So they call those connectivity fingerprints, the distinctive pattern of response of a given functional region of the brain to all other regions in the brain. Zeynep Sagan figured out how to look at these in humans using diffusion imaging, which we'll skip all the details and just say it's a not great, but best we have, method for looking at structural connectivity in human brains. You just follow vectors of maximal diffusion to try to infer long-range connectivity. And so what Zeynep did was she took every voxel in the bottom of the brain and measured the strength of its connection based on diffusion imaging to each of 85 other anatomical parcels in the brain. And she showed that that connectivity fingerprint of each voxel was strongly predictive of how face selective that voxel was. And so she showed that you can predict exactly where the fusiform face area will land in each subject from the connectivity fingerprint of each voxel in their brain without even looking at their functional data except to check your results. So she showed that first for the fusiform face area in an early paper with Rebecca Sachs, and then later she and I and others showed this for lots of other functional regions elsewhere in the brain, that they also have distinctive connectivity fingerprints. So that's all about the relationship between structure and function in adults. But what I want to propose is that we can consider this as a hypothesis about development, that these long-range connections don't just exist in adults, but they're maybe part of the causal mechanism for how the functional organization gets wired up in development. And so there's actually some evidence for this already. So the long-range connectivity of each cortical region seems to be mostly present at birth. A bunch of studies, not mine, argue for that. It also predicts the location of functional regions that arise later. So Zeynep Sagan and I showed that if you scan 5-year-olds with diffusion imaging and you get connectivity fingerprints of every voxel in the brain, you can predict exactly where that person's visual word form area will arise a couple years later when they learn to read, even though you're collecting the diffusion data before they have a visual word form area and before they can read. Okay? That's cool, but it doesn't show the causality. Actually, causality was shown way back before us in the famous experiments that Morgan Casseur did with rewired ferrets. And basically what he did was to surgically change inputs in developing ferrets so that inputs that would have normally gone to visual cortex got piped into what would have normally been primary auditory cortex. And the result was that that patch of cortex getting visual input took on many of the properties of visual cortex, including ocular dominance columns and orientation columns and the whole bit. And so that was a very strong causal test of connectivity determining functional responses. Okay, so that's all why this is plausible. So now the question is, can connectivity fingerprints explain the very similar locus of the face selective regions in blind and sighted subjects? Or put another way, the way we decided to test this was to ask whether the connectivity fingerprint, that is the connectivity, the set of connections to the rest of the brain that is diagnostic of face specificity, that you can use to predict face specificity, whether that fingerprint was similar in blind and sighted subjects. And so for this analysis, we used resting functional correlations instead of diffusion tractography. It's another imperfect, in fact, probably slightly worse method than tractography. They're both pretty crappy, but they're all we have, so we use them and hope for the best. And so we're calling these correlation fingerprints because we don't really know their connectivity. They're a proxy for connectivity. But we use the same basic idea. So essentially what we do is we look in the sighted subjects, and we look at the patterns of correlations between each voxel and every other voxel in the brain at rest, use that as a measure of connectivity, and figure out what pattern of connections is diagnostic of face specificity that we can use to predict face specificity. You do that first in sighted subjects, and then you take that connectivity fingerprint and ask how well it can predict haptic face selectivity in blind subjects. That's the idea. And it works. So just to show you, here's one example. This is a bottom view of one of our blind subjects. The white outline is the whole fusiform gyrus where we're making the predictions. And this shows you the actually observed face selective response haptically in this blind subject, like the data I showed you before. But now, for comparison, here is the prediction of where the face selectivity should arise in that same subject based only on their resting functional correlation fingerprints. Trained up from sighted subjects. So you use sighted subjects to predict, to get the connectivity fingerprint prediction, you apply it to this subject's resting functional correlation, and you get this prediction, and it's pretty good. In contrast, something that you might have thought would be even stronger is actually weaker. This is what you get if you take all the other blind subjects and do just a group analysis of where is haptic face selectivity in the blind subjects. And that's not near as predictive as this subject's own correlation fingerprint. Now that's just one subject, but we see that pattern in nearly all the subjects. So here's the strength of the prediction of where a blind subject's face selective region will land based on the cited connectivity fingerprint. And here's the strength of the prediction based on functional haptic face selectivity in other blind subjects. So if I lost you there, sorry about that, I crammed in a lot. The main point is we can predict the location of the face selective region in blind subjects from the correlation fingerprint derived only from cited subjects. And we can also do it the other way around. And so another interesting thing is it seems like those predictions of exactly where that face selectivity will land in the blind subjects come not just from connectivity, feed-forward connectivity to retinotopic, early visual regions, but also from correlations with higher-level regions in the parietal and frontal lobe. So the conjecture we're putting forth here is that it's top-down connections that instruct the location of the fusiform face area in the blind subject, and presumably also in sighted subjects. Now, there are heaps of open questions that we don't know the answer to yet, like exactly which regions are driving that connectivity fingerprint. I've mentioned before that neither of these methods is perfect, and I'm not sure I believe any of the individual correlations or connections, so that's a hard question to answer. A second important question is, what is a function of that blind FFA? Marina Bedney and Judy Kim have a beautiful recent paper where they show that what people thought was the visual word form area in blind subjects feeling Braille is actually a higher level language region. It's sensitive to linguistic syntax and stuff like that. So we don't know with our blind subjects here whether that's really a face area or whether it's subserving some other aspect of social cognition. And also, we don't have a good answer to why there's so much variability across the blind subjects, and heaps of other questions, just to say this is ongoing and unfinished. Okay, so where do we get with this question of how all this stuff gets wired up? Well, I argued first that face preferences develop very early in their characteristic location. They can develop in that location without visual experience, and maybe the cortical locus where that face selectivity will arise is determined by connectivity fingerprints that are present at or very shortly after birth. Okay, so now that we've grappled a bit with how these things develop, I want to move on to an even bigger and more difficult question, and that is, why do we have functional specificity in the cortex in the first place? That is, why, from a computational point of view, does it make sense to have modular design? Why don't we just have all the bits equally involved in everything? And in particular, further, why do we have the specific functional specializations we do? So in my lab, we've spent a lot of time looking for other functional specializations, and we found some others that I haven't had time to talk about today or show you. But it's not like we find cortical specializations for everything that might make sense. We don't find them for seeing snakes or seeing food or seeing spiders or seeing cars or lots of other things for which you might be able to tell a story about why we might have that. So why the ones we have and apparently not others? I always used to end talks right there, kind of acknowledging that that was the big open question and thinking, I'd love to answer that, but I can't even imagine how one would ever go at that. But very recently, we have an idea of how we might start to go at it. And so this is work with my postdoc, Katarina Dobbs, an important collaboration from Alex Kell. And we've been focusing first on why we have special machinery for faces. And the strategy, shockingly from my lab, since I have no expertise in this, is to use convolutional neural networks. So the basic hypothesis that we want to test with convolutional neural networks is that the ideal feature spaces for faces and objects are so different that one system just can't do both without a cost in performance. Okay, and that might be why we have special bits in the brain. And so lots of people have said stuff like that before. I've said vague versions of that hypothesis for a long time, but it was just loose talk without a way to test it. The idea here is that we can actually test this idea with convolutional neural networks. So recall that back at the beginning of the talk, I showed you Katharina's face-trained convolutional neural network based on AlexNet trained on lots and lots of faces. And I showed you before that this network, if you read out the feature space from the penultimate layer, it does above chance at object recognition. Before I was emphasizing that it's above chance, but now I want to emphasize that actually it's really bad. Performance is really low here. And that suggests that that feature space might not be very good for object recognition. Well, what about faces are highly specialized? Maybe an object recognition system is very general and would do fine with face recognition. So what about the opposite question? So Katharina then trained up AlexNet on object recognition using one of these massive ImageNet databases. I mean, here are things you learn when you start trying to train, have people in your lab training deep nets. It takes 2 weeks just to download ImageNet. Who knew? And then you have to buy all these expensive GPUs. It's a whole nother universe. Anyway, so what does Katharina find for the object network when you feed it faces? You get rid of it's only trained on objects, so you can't read out classification accuracy from the top layer. So you get rid of the top layer, take the features out of the penultimate layer, train a linear SVM. If you don't know what that means, don't worry about it. We're just trying to read out that feature space and ask, how good is it for face recognition? So here it is. It's great for object recognition and crappy for face recognition. Okay? So what this is telling us is that optimized feature spaces for faces don't do well at object recognition and vice versa. And maybe that's why we have different processes in the brain for face recognition and object recognition. But I haven't yet done the strongest test. All I did was say, if you seek for an optimized face space or an optimized object space, those spaces don't work well on each other's task. But what about if you try to train one network on both? Maybe that would enable us to find a common feature space that would work just fine for both. So that was the next challenge. And so what Katharina did then was to train this third version of AlexNet. This is a fully shared dual task network. And so what she did was first train it on a bunch of faces, then a bunch of objects, then a bunch of faces, then a bunch of objects, and you do that for weeks until the thing plateaus at performance. And now we can look at classification accuracy of all of these networks and compare them directly. We don't need to do that funny business with the support vector machines. So let's look first at the single domain networks, the face only and object only networks. So this shows you classification accuracy for faces in the face only network and objects in the object only network. They're both good. If you're worried that objects are low, I will just mention that that our performance of our AlexNet network is on a par with all the other reports of, actually slightly better than most of the other reports of object recognition in AlexNet. And if 53% correct seems unimpressive, recall that it's choosing 1 out of 600 object categories, and also the ImageNet database is not all that great. There's a lot of weird images in there and weird labels. And this is a percent correct of top 1, not top 5, as in lots of the machine learning literature. So that's actually very good. Okay, so now, key question, what about the shared network? How does it do compared to these single domain networks? And the answer is it does not do as well. So there's a very significant drop in performance for face recognition on the dual network and a very significant drop in object recognition performance in the dual network compared to the object-only network. And I think that means that, in fact, we do have a cost of sharing. You can't get the same network. You can't use the same feature space to do both tasks without incurring a cost in performance, supporting our hypothesis. Now, there's lots of other questions for ongoing work. So right now, Katharina is asking, well, what about, can we share some of the first few layers without incurring a cost? Maybe you can share the first layer and branch after that, or the first 2, or the first 3. So she's now training up a whole family of networks, asking whether maybe the first few stages can be shared before you need to specialize with a separate pathway for faces and objects, like we see in the brain. So this is all ongoing work, and I'll just say where we got with this so far is that it looks like face and object recognition may be distinct in the brain because there's no common feature space that can do both without a cost. Of course, we're not done yet. For this line of work to have any real explanatory force, we need to further show a similar cost of sharing for place recognition and object recognition, because those are separate in the brain, but not for car recognition and object recognition, which don't seem to be separate in the brain. And so we're working on those now, and we don't have those results yet. I should say that this line of work was inspired by a beautiful study that Alex Kell did with Josh McDermott, published last year, where they did show a similar kind of sharing cost for speech perception and music perception. Who knows, maybe we will eventually, once we figure out how to train up networks to do other aspects of cognition, maybe we'll be at some point able to explain a lot of this functional organization by which things can be done in a single network and which ones can only be done optimally when separated from each other. Maybe even someday we'll know how to look at whether theory of mind and language need to use separate networks. Okay, in closing, I want to re-acknowledge Katharina Dobbs, who did the deep network. I just want to emphasize that was quite amazing to do this in my lab. We had no expertise at all, and she really taught herself how to do all of this. It was quite remarkable. And by the way, she also does very beautiful empirical work using MEG and functional MRI and behavior. And she will be on the job market soon. And also Alex Kell, who had done the original study showing the cost of sharing for speech and music that inspired all of this work, and who's been an absolutely essential collaborator. And Ratan Murthy, who did the blind studies, along with important help from Santani Teng and a bunch of other people. And finally, I want to acknowledge my wonderful lab, who not only work really hard and know a lot of stuff I don't, but who are also willing to try a whole lot of crazy stuff with me. Thank you.